afternoon and welcome to the public webinar hosted by APC Microbiome Ireland, which poses the question, can we shape our microbiome with food and viruses? And on this rainy afternoon, we have two excellent speakers, which will hopefully make it a little less painless to be looking out at a dull, dreary November day. The first up is Dr. Susan Joyce. Susan is a lecturer in the School of Biochemistry and Cell Biology in UCC and also an investigator with APC Microbiome Ireland. Susan's main interest is in microbial systems which crosstalk with human cells. Her interests include modification of dietary components and host-produced molecules for responses and cross-communication in health and disease. And Susan's research has developed new connections with inflammation, body clock and weight gain. Susan is a partner on many international projects, including Kabbalah and Sophia, as well as national projects, FibreMet. And we will encourage you to enter any questions or comments that you have in the question and answers box, and we will take questions after each speaker. Enjoy the talks. Thank you, Susan.
So hello everybody, and thank you Orla for the wonderful introduction. I'm a scientist and a lecturer here at UCC, and I'm delighted to celebrate Science Week with you. My chosen title is You Are What Your Microbes Eat. And we've all heard the phrase, you are what you eat, but we know that, and this cartoon actually shows it, I'm sorry, here we go, that this isn't strictly true, okay? So I want to introduce you to the wonderful world of the gut microbiota, which we study here at UCC as part of APC Microbiome Ireland, okay? So this is our building here in UCC, and my group is part of the host microbiologue. We form part of a larger family or community that consists of researchers, academics, clinicians and consultants who all come together to answer and to ask pertinent questions regarding the influence of the gut microbiota on gut health and indeed in related diseases. So the gut microbiota is an umbrella term for a mixture of bacteria, fungi and viruses, and indeed bacteriophage, which Professor Hill is going to talk to you about in a while. It represents your very own co-evolved system, and it is with you from birth until death and everywhere else in between. It adds an extra repertoire of about 3 million genes to your own. And when analysed to the species level, and you carry greater than 1,000 species within you, it is clear that it represents a unique fingerprint for each individual, so that the gut microbiota for you is different from the person who would normally be next to you. So, in terms of this, we can categorise the microbes, in particular bacteria, into groups or to phyla. And here you see the 4 dominant phyla that are represented by bacteria in the gut. And these phyla are altered depending on the locations you go to in the body, whether that be the skin, the nasal passage, the vagina, the oral cavity, okay? So in the last 15 years, the field has advanced with the omics in technologies in terms of what we can do, okay? The omics have helped us to move beyond who is there in the gut to decide what they might be doing, to determining what they are actually doing, okay? And it's actually become clear to us that it is the redundancy and the functionality between ourselves and the microbes that is the convergent factor, at least from our point of view in the lab. So if we examine our representation going from birth to death across the lifespan, we can see that the microbiota can change, and this is dependent on a range of factors, including intake, who we are taking in, what microbes are colonizing us, the food that we eat, and therefore the food that our microbes are eating, and antibiotic use, so who we bring in, okay? And of course, our own host genetics and host factors contribute to the process. They're the genetics, our genes will determine a lot of what's going on in our interactions, our hormones, and indeed our immune system will shape the gut microbiota as well at different stages of life. So when we are feeding ourselves, we are also feeding our gut microbiota and our gut microbes. And if we provide a food substrate that is a particular set of microbes we can utilize, that a particular set of microbes can utilize, then they will bloom within the gut, okay? If not, such microbes may remain quiescent or at a constant level, or they may be lost over time. In providing a substrate from one group of microbes, And in their conversions, they may produce another metabolite or factor that can enhance the growth of a different group or indeed reduce it. In terms of this means that the gut represents a dynamic functional environment impacted by exposure by food type, by interactions and by host immunity. And we can detect these changes. And the best example that I can give you here is this study here of early life, where gut colonization is dependent on exposure to microbes, on mode of feeding, here we see with the breast milk, on weaning to solid foods, and on our dietary diversity, what we bring in, what route we take, what type of food we're going down the road to in the longer term. And we can, and by examining the genetics of these organisms that are represented here in these different Petri dishes, we can infer who is represented and if they are represented differently among groups or cohorts of individuals. And we can also, if the range of these microbes in these cohorts are actually altered, as you see here in this instance. Okay. So in terms of our work, we consider all of these elements important: the host factors, the microbiota, the diet, and they all converge on the functionality of the gut microbiota and how they can crosstalk and shape our gut microbes and impact our health. And we examine this in terms, as I said, of metabolites and metabolism. And our main parameter in looking at this is our fitness to function, and we frame this in terms of health and indeed disease. Okay, so one of the obvious representations where host factors, food, and the gut microbiota come together is in considering differences to body mass index or to BMI here. Okay, this is a real issue in the Western world and is associated with comorbidities, including cardiovascular disease, type 2 diabetes, colorectal cancer, and indeed projections from WHO, the World Health Organization in 2008 indicated that levels of obesity would reach 40% among Irish males and 37% among Irish females by the time we hit 2020. And I'm not sure whether we've reached those parameters or not, but this actually induced the government to bring in certain measures and to, I suppose, finance certain areas of research. So, a clear role for the gut microbiota in BMI had been established, and the most concrete form in transplanting microbial populations from high and low BMI individuals to mice that were lean mice but fed the same diet. Those who received the microbiota from high BMI individuals gained more weight, and they gained it progressively. Okay. However, in considering this, classically we associate high BMI with long-term positive health, energy balance, so that extended calorie intake over calorie utilization over time will result in a higher BMI. And this is the model that we've always been working on. However, high BMI is not necessarily unhealthy. And this was shown by Orla O'Sullivan's group in 2017 in a study comparing the high BMI athletes to low and high BMI individuals in the normal population. And what they were able to show was that the gut microbiota of the athletes more closely resembled that of low BMI individuals in terms of representation and in terms of diversity. What they were also able to show was that the functional differences were present among those individuals that were athletes of low BMI and of high BMI, and that in general the athletes associated with those low BMI individuals, and this is represented by the colours and the differences in the colours that you see here. Okay. We now accept that microbially produced or altered factors can elicit their effects at the level of the gut epithelia and indeed systemically, and that they influence outcomes that can result in either health and disease. And some of these factors are listed here, and among them are bile acids, which are of particular interest to my group. These are not a newly discovered set of molecules. In fact, they have been used in traditional Chinese medicine for hundreds of years, and they're claimed to cure nearly all ailments. But here is bile as we receive it in the lab, and it contains a number of different components, including proteins, lipids, pigments. But the major component, and the component that is really of interest to us, is bile salts, which represents about 2/3 of the composition of bile. Now, what's interesting is that bile salts are made from cholesterol in the liver, and they're emptied and stored in the gallbladder for postprandial release, so released after you've eaten. Okay. And we know and we recognise that bile is an emulsifier of fat, and in doing so, it releases those essential vitamins that you cannot make yourself, the vitamins D, E, K, and A, the DECAs. It keeps bacterial numbers in check, and as a result of this, bacteria have evolved wonderful and elaborate means in order to counteract its effect. It has enjoyed a resurgence in popularity in the last number of years because it's now recognised as a signalling molecule for both endocrine and immune functions. And this is what we concentrate on here. Now, in looking at the circle of bile through enterohepatic circulation, you can see that bile, while it's made in the liver and released into the small intestine, 95% of it is reabsorbed by the time it hits the lower part of the small intestine. And only 5% goes through for excretion in the feces here. Okay, this is one of the only ways in which we can eliminate cholesterol from the body. But not only that, this bile can act as a signal going from the gut to the liver, which processes most of our metabolites. Okay, and it can redirect metabolism in different directions. Okay, so I'm going to look at the signaling that's going on here. I'm going to tell you a little bit about that. Okay. Right. But first, what I want to tell you is that bile has a signature, okay? And this signature is altered as we move through the gut. So, while we can look at gallbladder bile, by the time we reach the stool, where all of this has gone through the intestinal tract, that signature has changed completely. And the blood and the urine has its own bile acid signature as well. Now, this signature changes depending on the reuptake systems that are activated, those transporters I told you about in the small bowel region. It's also altered by feedback signaling to the liver, and it's also altered by the resident microbiota. So how do they do this? So it turns out that the vast majority of gut commensals are equipped with an array of enzymes called bile salts hydrolases that can modify bile acids differentially. They're the gateway reaction for modification by other microbes. And these modifications produce what we call a palette of signaling acids here, or bile acids here. And this is just a subset of them. They go up into their hundreds. They signal not only to microbes, but also to your own cells to mediate and modulate processes, including metabolism, energy homeostasis, inflammation, and toxin clearance to cells that contain these receptors. Okay, and this is important. And we wondered whether we could reset or interfere with these signals through microbial intervention. And the quick answer to that is yes, we can. We can do that. Okay. So we can dampen and reset these signals. And I'm showing you this. We see this in the situation of recurrent C. difficile infections and fecal microbial transplants. Okay. Recurrent C. difficile infection occurs as a result of overuse of antibiotics, generally in elderly patients where first infection occurs. What we were able to show in this instance here was that the patient microbiota, or marked here in this red circle here, okay, is distinctly different and altered in comparison to healthy donors, those that are marked D here. Okay. When FMT is given, the microbiota recovers somewhat towards the healthy donor profile. On a metabolite level, we saw that bile acids that normally maintain C. difficile in check in a dormant state were reduced in these infected individuals, and that fecal microbial transplant restored this balance and functionality to the microbiome. However, a word of warning, it's not it doesn't cure all ills, okay? Not every function was reset and restored, however. We see that in terms of the fatty acids that we see here. We next investigated whether we could alter these signals through a single gene function of the gut microbiota, reducing it to just a minimalistic approach. To test this, we used the murine DIO, or diet-induced obesity model. We used a mouse model. We cloned 2 different high activity BSHs, one with high activity, one with low activity. The bile metabolizing enzymes, and we fed them to mice that were germ-free with a gut colonizing bacteria, an E. coli enzyme here. And here you can see in this mouse that the light-producing bacteria, which are labeled MG1655, are actually colonizing the whole of the intestine here. For the first time, we were able to show that bile acids impaired a number of functions in the gut, including lipid homeostasis and signaling, transporter function, immune function, and indeed it could alter your body clock or your circadian rhythm, that clock that tells you when to sleep, when to eat, when you're at your best in terms of learning and working. Okay, we could adjust that through bile acid alterations. When we went back and we recolonized conventional animals that are those that are raised with a microbiota, we saw that we still had a high colonization, but what we saw in these animals was that we were able to reduce weight gain. And this was reflected at the level of serum cholesterol, but also in terms of insulin resistance and in terms of glucose clearance as well. Okay. This opened up a number of different areas of research that we were able to confirm, where we were able to confirm a role for microbially modified bile acids. When we examine different states to see where microbial populations are altered, so too are bile acids. And this is shown in this slide here. And these are the publications where we've looked at obesity, we've looked at C. diff infection, we've looked in the autism spectrum, and we've also looked at short bowel syndrome and a model of it in a porcine model. And we see what's happening here, that these signals are adjusted, and they're adjusted through microbial intervention and bile acid signaling. Okay. And we therefore considered bile acids as a good representation of the gut microbiota's fitness to function. And in terms of this in the lab, we have isolated a number of potential probiotic strains. In terms of their fitness and functionality, we examine them. We use microbe modified host bile acids through and also through modification of dietary fatty acids. We apply mass spectrometry to analyze our systems. We can then draw up a heat map that reflects the activity of each strain. So we can match the deficits in different disease states with the strains and the outputs that we have in vitro. And we can assess the host responses in simple or more complex models. And that's the model that we're working on at present. So more recently, we have examined this in the context of health benefits of fermented foods. And here we've isolated Lactobacillus plantarum strains, which are known to carry up to 6 copies of bile salt hydrolase enzymes. We type them to strain level, we put them through European safety standard guidelines and testing, and we examine their ability to metabolize bile acids. And in terms of this, what we were able to see was that among the majority of these foodborne L. plantarum isolates was that 2 specific modifications to bile acids were evident almost everywhere across the board relative to controls that were taken from other sources. First, we saw that these bile acids could dampen inflammation, and we could see that they did this first by maintaining the levels of a bile acid capable of activating the vitamin D receptor, and secondly, by activating a receptor present on macrophage cells that's known to dampen the immune response. So I hope that I've provided some flavour of how food, microbiota and lifestyle can modulate and shape the gut microbiota. And I'd like to thank you for listening, and thank the members of my team and also my collaborators within APC who are listed here. So thank you very much for listening, and also thanks to the funding bodies that are listed here as well. Without you, our work wouldn't be possible. And I'm happy to take any questions going forward.
Thank you, Susan, for a fascinating overview of the importance of the metabolites that our microbes produce. People seem to be a bit shy with questions at the moment, but I'll start off with one. Is there a particular type of diet that helps promote the production of these bile acids?
Okay, so I mean, different diets, the high fat diet will actually induce more production of bile acids and influx into your system. And there's a bit of a paradox with this, because you'd imagine that if this was the case, that in a high fat diet situation, that if you're removing some of the lipids and incorporating them into bile acids and pushing them through the gastrointestinal tract, that this would be a beneficial thing, but it isn't. In fact, this is what happens during bile acid malabsorption, causes bile acid malabsorption and diarrhea. But there are diets that will push through more. There are others, such as high fiber diets, which are very beneficial, which will actually bind up the bile acids and take them directly for excretion. So this will reduce the bile acid pool and therefore your cholesterol levels in turn. And as such, it will cause further cholesterol to be pushed into the synthesis of bile acids and be rebound again. And this is one of the mechanisms proposed for how dietary fibres can bring health benefits to an individual.
Thanks, Susan. And I suppose another question for me is, I suppose we all associate bile with the gallbladder, and I may have missed this, and apologies if I have, but what happens in people that have had the gallbladder removed?
Okay, so in individuals where the gallbladder removed, it's not very well documented. So instead of a gush or a flow or a regulation of release of bile in with your gastric juice and with your stomach contents on to reach the small intestine all at once, then in that instance here you would have a specific consistent release of bile acid or flow of bile acid directly into the small intestinal tract. And in fact, you know, for people who've had those removed, this is not so unusual, because in fact horses and rats don't have a gallbladder either, and they're just as healthy, so it's not going to impact your health too much. But where bile acids play a role, which is in gut transit, in motility, in their signaling effects, in the modifications that occur, I guess what you would say in that instance is that perhaps it would provide an opportunity for other microbes that would normally be kept in check by the high concentration that would arrive in the small intestine at a particular moment, or in the large intestine when it becomes concentrated later on, that it would provide an opportunity for outgrowth in this. And this hasn't been actually looked at in terms of that. So that's an interesting question, and it's one that should be followed.
Great, thanks, Susan. Just a couple of other questions. Have you linked specific primary or secondary bile acids to weight gain or weight loss?
Well, we can say if we've looked at a porcine model, and what we've started to do, and what researchers in general have started to do over the last few years, is to see what's happening over time. So what's happening from the beginning? So if we start eating a high fat diet, or one of the Western diet that promotes high BMI, then what's actually happening here? And what's happening in terms of the metabolites? And in terms of this, what we've seen in a porcine model, where we've looked at a porcine model, and where we've looked at murine models, and when we've looked in humans, is that we see an increase in, yes, certain secondary bile acids over time. And we see that some of these accumulate. But that doesn't necessarily mean that at the cellular level that this is a bad thing. Some of these are beneficial. And as I said, besides vitamin D, which we hear an awful lot about, you hear it in the news, you hear it in many different papers, you see that it has essential roles in proliferation of cells. It allows those cells that should be how will I put it, culled from a population to be culled, and I'm talking about your own cells in apoptosis. It has many different benefits. And the only other component besides sunlight activated vitamin D that can actually activate this vitamin D receptor to perform all these functions, including toxin clearance, is actually the bile acid LCA. And we see that accumulate in a high fat diet situation over time, and we see it gradually rise over time. Now, we do think that because it's highly hydrophobic, that eventually it reaches a level, and it's normally cleared. It's normally cleared, okay, from your system. It induces vitamin D receptor. Vitamin D receptors allows it to be labeled, and it's normally cleared out of the body. But in the instance of a high-fat diet, your transporters become altered, and this one actually accumulates and makes its way back to the liver. And in terms of that, it can do damage then, and that's when the problems arise. So when the system breaks down, you're okay to a point, and then everything just goes over a threshold. And that's our opinion on those in high-fat diets and in obesity.
Great. There's a few other questions, but just in the interest of time, I'm going to take one more question. What effect does dietary cholesterol play in bile acid production, if any? Okay, so dietary cholesterol, there's actually two different schools of thought on this. Okay, so one thought is that bile acids maybe need to be synthesized from newly made cholesterol, because you can make cholesterol yourself. And cholesterol, you know, you need it. You need it for the structure of your cells. You need it in the body. You need a certain amount of fat. Cutting out fat is not a good thing in terms of your diet. But when you need it, you can also synthesize it yourself. And it's that synthesis process that actually your statins will target to get rid of your cholesterol, to reduce your cholesterol, but not get rid of it, because you still need to take some in. You still need to have some in order to provide some rigidity to your cells and some structure to your cells and for other processes. So in terms of that, the emptying of bile, of cholesterol into bile, and bile has really got, our cholesterol really has three fates. It's either converted into these steroid hormones, the sex hormones, etc. Okay, it's either converted into bile acid, and in that way it can be emptied or recycled, or it can be deposited within your cells. Okay, they're the fates, and it can be used as well to make vitamin D. So it's used for that, and it's deposited within your skin. So in terms of bile acids, they can have a large effect, and this has been shown in animals with certain strains of probiotics that can modify bile acids, but that can also induce the production of cholesterol by these modifications that cause these bile acids to be emptied more rapidly from our system. So there is that connection between them, all right. The other side of this is that both are taken together, and we don't eliminate these bile acids, and the signals aren't returned in the correct way, is that it can lead to a buildup of both lipid, including cholesterol, and bile acids in certain tissues, particularly in the liver, and this can lead to scarring, other injury, and other diseases. Thanks, Susan. And there's many other fascinating questions, and I apologise we don't have time to get to them all. We may have time at the end and we can go back to them, but in the interest of time, I'm going to Professor Colin Hill. And Colin is a Professor of Microbial Food Safety in the School of Microbiology in UCC, and he's also an investigator with APC Microbiome Ireland. And his main interests are in infectious disease and of late in elucidating the phagome of our guts. Colin has more than 570 publications and is listed as a highly cited researcher in 2018 and 2019 by Clarivate. He's a member of the Royal Irish Academy, and recently he was recognised by UCC with a prestigious Career Achievement Award, which celebrates exceptional research over a career. And Colin today is going to talk to us about bacteriophage and their role in the gut. Thanks, Colin.
Thanks, Orla, and thanks for the introduction. Okay, so I hope you can see my slides now. What I'm going to do is talk about viruses. Now, I imagine that most people are really sick and tired of hearing about viruses. Every time we turn on the radio or the TV, we're talking about this character. And I think we're all used to the fact now that viruses have a very significant impact on human health. And Peter Medawar maybe expressed it best when he said a virus is a piece of bad news wrapped in protein. And the bad news is the genetic content of the virus. And normally what viruses do is they enter human cells and they begin to replicate using their genes and using their gene products and produce multiple copies of themselves. But what you may not be as familiar with is the fact that bacteria also have viruses, bacterial viruses. And these are called bacteriophages. And here on the right, you see an actual picture of a bacteriophage. It's generated by an electron microscope. And you see the DNA is here within the head of this particle. The green bit here is a protein that surrounds the DNA. Then you've got these very elaborate tail structures that recognize the bacterial cell, and this kind of needle, which you see here, which injects the DNA into the target bacterium. So they're really pretty little molecules or pretty little microbes, but they can only, just like viruses, they can only replicate in the presence of their target. But as I said, in this case, the target are bacteria. So if we look at the life cycle of these bacteriophages, what they do is these receptor molecules on the bottom of the phage binds to a bacterial cell. So this green oval shape here is a bacterial cell. It injects its DNA, and the DNA immediately takes over the cell and begins to produce copies of itself, but also new phage heads, new phage tails, hundreds, if not even thousands of copies of these individual proteins and its own DNA. And then these viruses assemble within the cell, they form up. Every one of these now encapsulates its own DNA again. And then the last step of the viral infection is you get the programmed bursting of the bacterial cell and the release of these phage particles, these new infectious agents. And you can get between 100 and 200 new phage particles within about 30 or 40 minutes of this infection. So in the space of a few hours, one bacteriophage, one bacterial virus can become millions, much faster than bacteria can grow, and therefore they end up killing most of the bacteria in their environment. So we actually live on a bacteriophage planet. If aliens were to come to Earth and scan the planet and report back to their masters, they would say it's yet another phage or viral planet. There are about 5 by 10 to the 31 phages on Earth. There's more bacteriophages than any other biological entity. You could take all the microbes, all the bacteria combined, and yet we have more viruses, more bacteriophages. And this is what 5 by 10 to the 31 looks like, this number you see here. Just to give that some perspective, this is the number of humans in red on the planet. So there are trillions and trillions and trillions of viruses for every one of us. And they've been here for about 3 billion years. We've only been here for a couple of 100,000 years. So they're really well evolved to live on this planet and to dominate this planet. Now, they were discovered about 100 years ago, in fact, in 1917, by this guy here, Felix Durrell. He was a Canadian scientist, but he was working in the Pasteur Institute in Paris. And he published a book in 1921 on the bacteriophage and the bacteriophage and its behavior. And he was the one who came up with this name bacteriophage. And when he was working with bacteria in his lab, he saw that every now and then you got this kind of hole in the bacterial colonies. And he said there's something eating the bacteria. So he called them bacterium eaters, or bacteriophage from the Greek for eating, phagein. And so he was very interested in what was happening here. What had landed on this plate that was killing these bacteria so efficiently? Now, he collaborated with another guy called George Eliava. Eliava was at the Pasteur Institute, but he was actually a Georgian at that time in the Soviet Union. And Durrell and Eliava, you see them working here together, went to Georgia and they set up this phage institute called the Eliava Institute, which still exists this day. This is a modern day photograph of it. And they set up huge systems for producing these bacterial viruses. And of course, their objective was to use them to treat disease. If a bacteriophage can kill a bacterium, then maybe they could kill the bacteria that are causing infections in humans. And as I said, this institute is still viable about 80 years after the event. Now, Eliava, unfortunately, didn't have such a prosperous time when he went back to Georgia. Eliava was a bit of a ladies' man, as you might be able to guess from this photograph of him here. And he had an interest in this lady, Tinatinjikia. But unfortunately for him, Laurenti Beria, who was Stalin's chief of secret police, also had an interest in Tinatin. And so Beria arranged for Eliava and his wife, unfortunately, to be executed. So he was executed as an enemy of the state. The institute went into decline for a while, but it did make a recovery, and it still exists today. So this is what you see if you go onto the Eliava Institute website right now. They produce viruses, they produce them in very large amounts. and they produce them for this pharmaceutical use. So you can buy phages, you can buy bacteriophages, bacterial viruses in Russia, in Georgia, to actually treat disease. And here's some of the things that you could buy in a pharmacy. So this one's for treating intestinal diseases, this one for skin diseases, for lung diseases, and so on. So you can buy the various different phages you might need to treat your disease. There are some problems with phages, and I'm illustrating two of them on this slide. One is specificity. Most phages only kill a very small number of bacteria. So here you see this phage down here, for example, phage C. It can kill bacteria number 2, but it can't kill bacteria 3 or 1. So if you're infected with bacteria 1 and you try and use phage 2 to treat it, obviously it's not going to be very successful. And so this specificity was always regarded as a very big problem. Nowadays, of course, we like the idea of specificity because it means we won't do damage to the microbiome that Susan was talking about, and we want things that might go in and very specifically kill the target species. But it is a problem in terms of picking the right phages. And the other problem is phage resistance. If you plate bacteria on a plate that already contains lots and lots of phages, some bacteria will still grow. And when you grow those up, you find that they become resistant to the phage, somewhat in the manner in which strains can become antibiotic resistant, they can become phage resistant. So you have these two significant problems. So what the Eliava Institute do to overcome these problems is they use hundreds of different bacteriophages in each vial. So this is a vial of bacteriophages that would be used to treat skin infections, for example. And you might think that it contains just one virus, but in fact it contains hundreds of viruses. And as new bacteria appear that cause skin problems, they just keep adding in more viruses. So they keep adding in more bacteriophages as new infectious bacteria appear. So the vial you buy this year might be different to the vial you buy next year because more phages have been added, maybe some phages have been removed. And unfortunately here in the West, the Food and Drug Administration in the US or the European Medical Agency, they do not approve medicines like that that can change year to year. They want them to be very specific and very defined. Also, they don't really conduct very good randomly controlled trials in Russia to get clinical approval, as we've seen with the COVID vaccine. The rules are different and they're not really accepted by most Western regulatory agencies. I might wonder nonetheless why we don't have phages in our pharmacies, and that's because the West, as I call it here, really went with antibiotics. Antibiotics were a miracle drug. They're broad host range usually. One molecule can kill nearly all bacteria, and so they're very useful. You don't have to worry about what the infection is when you're picking one of these antibiotics. And they were discovered by Alexander Fleming, who discovered penicillin, which is the molecule we see here, and Selman Waxman, who discovered streptomycin, which is the molecule we see here. And of course, we've discovered many others since then. These two got the Nobel Prize separately for their work in finding these different antibiotics. And antibiotics really changed medicine. This, in 1938, was the treatment for tuberculosis. And tuberculosis, of course, was an extremely common disease here in Ireland, in the UK, everywhere. And here you see that the treatment is the fresh air treatment. Just simply pushing people's beds out into the fresh air during the day, moving them back in if it started to rain, back out again, and back in for the night time. And here you see beds just close to London Bridge, across from the House of Parliament, with the patients sitting out in the fresh air. And that was essentially all we had. This is one of the very first people to be treated with penicillin in 1942. This young girl had a very nasty infection that would have almost certainly killed her if she'd been born even a year or even a few months earlier. But she was treated with one of the very first batches of penicillin. And what you can see here is that within a week, she started to make a very significant recovery, and within 2 weeks, she was completely recovered. Nowadays, we might regard that as something pretty routine, but this was spectacular at the time. In fact, the penicillin was so valuable that they collected this girl's urine and repurified the penicillin and used it again over and over because it was so valuable. And penicillin alone, it's believed, has saved about 200 million lives in the 80 years we've had it. And all antibiotics have saved about 400 million lives, probably the greatest medical invention of all time, with the exception probably of vaccines, which are equally good and maybe have even saved more lives. And they were so good that in 1969, William Stewart, who was probably the 60s version of Anthony Fauci, said the war against infectious diseases is over. Antibiotics are so good, we don't have to worry about infectious disease anymore. And he's sometimes portrayed as a bit of a fool or an idiot for having said that, but at the time he was absolutely correct. It seemed that with more and more antibiotics and we could cure virtually everything with them. But of course, as you all are aware, we now are in the era of the antibiotic-resistant superbugs. So antibiotics were really abused. They were not always used appropriately. They were overprescribed. They were used for viral infections. Many, many more antibiotics were used in animal welfare than in human medicine. And a lot of bacteria. This is the same bacteria a few years apart. And here you see the bacteria is killed by every one of these different antibiotics. And now it's only killed by a small number of them. And some bacteria have now appeared that can be killed by none of our antibiotics. And bacteria become resistant by mutating, but also by sharing these resistance genes among themselves. And so it's a very big problem and one that we're really struggling with. And in the middle of this pandemic, we can already see what the next pandemic is going to be. The next pandemic is going to be antibiotic-resistant superbugs. So to put it in some perspective, COVID so far this year has killed almost 1.25 million individuals, which of course is absolutely frightening. But by 2050, it is estimated that more than 10 million people a year will be dying of untreatable superbugs. Bugs like this one again, that used to be sensitive to all of these antibiotics and now is only partially sensitive to one or two. So we've really almost blown our antibiotic pipeline and we need to develop more and different solutions. And so now we're starting to turn our attention back to viruses and back to bacteriophages. So are we entering the new age of phage? The next 100 years is going to be a return to looking at phage in Western medicine. The advantages are pretty obvious. There are trillions of different phage on Earth. We'll never run out of phage. We might run out of antibiotics, but we'll never run out of phage. They kill bacteria very efficiently. They cause the bacteria to actually burst, so they really kill it. They multiply at the site of infection. So long as you can get a small number of phage to the site of infection, they'll multiply and kill all the bacteria at that site. So we don't have to worry about dosing as much as we do with antibiotics. And very importantly, they do not cause damage to non-target bacteria. So the virus will only kill the bacteria you're targeting. They won't cause damage to the microbiome. Or they won't cause other bacteria to become phage resistant and start to spread that resistance. And a lot of companies now are starting up and have been in existence for about the last 10 years or so in the West that are looking at phage as molecules that we can use in human and animal medicine. And this is just a very small array of some of those companies. So let me give you a couple of examples. We can use phage in the West, but we can only use them in compassionate use. In a case where nothing else has worked, an individual doctor can choose to use a phage. So here we've got Thomas Patterson. Now Thomas Patterson picked up an infection with this bacterium, this multi-drug resistant bacterium, Acinetobacter baumannii. He actually picked it up in Egypt while he was traveling. He came back to the US, but he fell into a coma, and the doctors gave him essentially no chance of recovering from the coma. He was resistant, or this bacterium was resistant to all antibiotics. Now, luckily for Thomas, his wife is Stephanie Strathdy. She's an infectious disease epidemiologist working in San Diego, and she had known about phage, so she actually tweeted to some of her colleagues, does anyone have phage against Acinetobacter baumannii? Someone came forward with some phage they had in their lab, and the doctors agreed, because Thomas had no other options, to infuse him with this bacteriophage. And this is Thomas now with Stephanie, and they've written a book on this perfect predator. He made a complete recovery. Of course, from a case just involving one patient, we can't make too many conclusions, but it does show that potentially phage could have a very good impact in medicine where antibiotics fail. Just give you one other study. This one was in England. This is Isabel Holdaway, a 17-year-old girl who had a lung transplant. She then became infected with this bacterium called Mycobacterium abscessus, and this one also is antibiotic resistant. And she reached a point where the doctors gave her less than a 1% chance of survival. So once again, in this case, the doctors went looking for bacteriophages, and they put out the word that they were looking for them. And Graham Hatfull, a US scientist, came up with a combination of 3 different viruses. He tested over 15,000 vials against the specific bacterium that was in his Isabel and selected 3 for treatment. Now, this took months to do. Now, obviously, you couldn't do this with a disease that is going to kill someone within a matter of days. But Isabel was in a fairly stable state, even though she had no real chance of recovery, and she made a perfect recovery after being administered phage. So we're also working on phage at APC Microbiome Ireland. We're very interested in this disease, cystic fibrosis, because people with cystic fibrosis can suffer very severe lung infections with this bacterium here, Pseudomonas aeruginosa. And this can mean that a person with cystic fibrosis can be on antibiotics essentially every day of their lives or require multiple treatments every year. So we badly need new therapies because these bugs are becoming antibiotic resistant. Now, we know, of course, that our gut is full of bacteriophage. Susan already mentioned that. This is just a microscopic view of our gut where the phages have been lit up with this green color, and you can see all the phages present in a gut sample, and they come in all different kinds of shapes and sizes. So we went looking in the gut for phages against Pseudomonas aeruginosa. This work was done by Debebe Alamayehu and Paul Ross, my collaborator in all this. And what we did was we looked for and found 2 phages that could kill a huge range of Pseudomonas strains. We purified these in the lab, and then we tested them in this model of cystic fibrosis infection. So what we've got here are human lung cells growing in the lab, and we've infected them with Pseudomonas aeruginosa. And that's the color, the bright green and red you can see here is the Pseudomonas aeruginosa. And then we treated the bottom well here that you see on both sides with the phages. This was at T0, and within 24 hours, we completely eliminated all of the Pseudomonas aeruginosa without causing any damage to the lung cells. Whereas the ones that didn't get administered phage, the Pseudomonas aeruginosa were still growing and causing damage. So it just gives a very quick example of how these phages can work in these kind of settings. Another disease we've taken on is another one that Susan's mentioned, Clostridioides difficile associated diarrhea, excuse me, CDAD. This is very, very difficult to treat with antibiotics, partly because it's a spore form. You can see the spore here. And unfortunately, there's about a 10 to 20% fatality rate. And here you see the gut of somebody with Clostridium difficile associated diarrhea, and you can see that it's in a very bad shape. But unfortunately for Clostridium difficile, no phage have ever been isolated in the laboratory. And so we also weren't able to isolate any phage to kill C. diff. So we took another option. This time we went back and looked at these enzymes called phage lysins. One bit that I left out earlier on when I was talking about the phage life cycle is that another thing that the phage produce when they start to replicate within a cell is they produce these little enzymes, and these enzymes then chew up the cell wall, and that's what releases the phage at the end. So these are very specific, very active enzymes that can also damage bacterial cells. So what Shakan Mandal in our lab, together again with Paul, he went and found a gene that encodes a Clostridioides difficile phage lysin. This is the enzyme, the protein, the Clostridioides difficile lysin, and Shakan purified it in the lab. He cloned it in E. coli and purified it. And here's 2 tubes, each containing high numbers of Clostridium difficile. He's added phage to the right-hand tube, and within minutes, 10, 15, 20, 25, 30 minutes, this tube has gone completely clear. All the C. diff within that tube have been lysed. They've all been killed. You can see this as well in a graph on the right. So we think the phage lysins will also be very useful weapons in our fight to overcome these antibiotic-resistant superbugs. So what do I think is going to happen in the next 100 years? Well, we have to take advantage of these bacterial viruses They're the most abundant and diverse microbes on Earth, and we must be able to use them to human benefit. Our gut is full of bacteriophages. Now, I didn't talk about this aspect of our work, but we know that bacteriophages also shape our microbiome and therefore influence human health. We very urgently need new solutions for superbug infections to prevent this next pandemic. Vaccines are not going to be any good against this next pandemic, so we need novel antimicrobial therapies. And phage therapy, I think, is one exciting option, but also these phage enzymes offer another therapeutic option. So if we live in this phage planet, we should be finding ways to use them to our advantage. So I leave it there. I thank Science Foundation Ireland for their funding, APC Microbiome Ireland, of course, that hosts all this work together with UCC. These are the people whose work I used in this talk, and Amy, Catherine and Mark, who set up this public webinar, which I was very pleased to take part in. So thank you very much.
Thanks, Colin. I have to say it's great to hear about a virus other than coronavirus for a change. So, and there's a couple of questions coming in. I think you may have answered a couple as the talk went on, but I suppose the first one, which probably is at the tip of everyone's tongue, has there ever been a phage that can fight another virus? You know, I suppose, can a phage fight coronavirus, for example, or any other virus?
Colin, you're muted there.
No, none has been found. There's a big debate over whether viruses and phage are actually alive or not. It's kind of a semantic debate, really, but they can't metabolize, they can't generate energy by themselves. They rely on the cell that they infect to generate energy and replicate themselves. So a virus has no energy and no way of generating energy, and a bacteriophage has no energy and no way of developing energy. So the two together can't really, one can't infect the other. There's nothing to grow on. There's nothing to really replicate on. So no, that doesn't exist as yet. Of course, science has a great way of surprising us, but I would be very surprised if that turns out to be the case.
Are phage lysins also strain specific?
Yes, and that's fantastic. I mean, we can add a phage lysin to something like a very complex bacterial situation like the microbiome, and the phage lysin just seeks out the target bacteria and kills it, but has no effect on any other bacteria. So they are just as specific as their phages, which is a fantastic property.
Great, thanks. One other question is, how far away are we from using phage in nutritional products, do you think?
They're already being used. Phage are being used in the US in food safety applications. So there are two different phage preparations that manufacturers can buy in the US, one that kills Listeria monocytogenes and one that kills Salmonella. And what food manufacturers do is they spray the vulnerable foods, like a chicken, for example, with the anti-salmonella phage. And then if any salmonella get into the chicken, the phage will kill them. But that's not really using them as a nutrient, if you like. It's using to protect the food and to enhance food safety.
Thank you, Colin. Do bacteriophage change if you exercise or change your diet?
Yeah, they do. We all have. What's been strange about phage, something we didn't expect really, was when we sequence the phage inside a person's gut, we find that each person has tens, maybe even more, tens of thousands of individual phage, and we share very few with each other. So you and I share almost no phage, probably absolutely no phage, but that's, so there's tremendous diversity. But if you do anything that changes your bacteriome, your microbiome, like diet, exercise, medication, you get a disease and it changes your microbiome, then of course the phage change too, because that's what they live on. So we do see changes in the virome when we see changes in the microbiome.
There's so many great questions coming in. It's hard to choose ones, and I apologize if your question isn't answered. I have another one here. Is, could phages help reduce pathogen contamination in the environment? So say, for example, to reduce nosocomial infections.
Yeah, it's a very good idea, and it's one that we're considering. I mean, it's difficult you'd have to spray the phages onto surfaces, if you like, and they probably won't last very long on surfaces. You probably have to keep reapplying them. they probably more of an option in things like food, as I said, or maybe in a surgery or in a clinical setting. I don't think they'll ever be used like in households or in food factories or anything like that. It's probably too difficult to use them there, but maybe in very limited circumstances they could be.
And this question links both talks. I guess, could bacteriophage be used to target specific gut microbes, which related to things like obesity, in order to promote the beneficial growth of microbes?
Yeah, and that's our dream. That's our real hope. You know, infection is very important, and that's the topic that I talked about here. But as big an aim for us is that when people like Susan and other people working the APC, if they can identify a bacterium that we think is having a negative impact on human health, what we'd like to be able to go in is go in and deliberately just knock that one out without damaging the rest of the microbiome. So we want to kill, you know, pathogens, but we also want to kill bacteria that are linked to poor health outcomes.
Great. Yeah, I'm struggling to choose which questions, because we really have so many fascinating questions that I would never have thought of myself. And I guess in the interest of time, I'm going to ask one more, and this probably covers a few questions. Is there a possibility that a bacteria could become resistant to a phage that it's being targeted for?
Yes, unfortunately, that happens very quickly. When you normally, if you expose, say, 1 million bacteria to a bacteriophage, the bacteriophage has to spread through that 1 million bacteria, killing them all very quickly. But usually out of every 1 million, you get about 1 that will be resistant. Now, sometimes it's 1 in 10 million, sometimes it's a bit less, but resistance happens quite quickly. And that's why the Eliava Institute and the Russians, they use hundreds of phages in each vial, because the chance of becoming resistant to all 100 is nil. So that never happens.
And I did like, because there's one more question that I'm going to ask you, Colin, and does diversity or do the profile of your phage change over your lifetime?
Yes, when you're born, you have essentially no phage, and they accumulate into vast numbers, and then they change over the course of your lifestyle or your life cycle, and they're different at all stages of life, we think.
Thanks, and we're going to, in the interest of time, we leave it there. Again, apologies, there was amazing questions came in there from everybody, and apologies that we couldn't get to them all. I'd like to thank everyone who attended the webinar, and to Susan and Colin for fascinating talks, and to Mark, Catherine and Amy in the background for arranging all of this. And I will encourage you to go on to the Science Week website and have a look at the rest of the amazing stuff that's going on until Sunday. Because you know, it's great to embrace science from home, even in a pandemic times. And again, thank you for everyone. And that concludes our session.